Hi, my name is Lea Yiannikouris, and today I will be talking about the elevation of fat mass mediated by human sPRR, and it is sex dependent. So to go over what I'm going to be talking about throughout my presentation, first I'll give a little introduction and background about what obesity is and what sPRR is. Then I'll give my research goal and hypothesis, my methodology, my mouse model validation, my results and data from my experiment, the conclusion, and some future prospects, and then some acknowledgements. So first, as a little bit of background, 1 of 3 American adults is obese and hypertensive. And more locally in Kentucky, Kentucky is ranked 8th in the highest adult obesity rates and 3rd for the highest youth obesity rates, which is an astonishing number. And I want to figure out why. So obesity is an increased risk of cardiovascular diseases, including hypertension and death. So it's a pretty severe disease. And obesity activates the renin-angiotensin system, or the RAS. And this is basically just a system that cleaves angiotensinogen, something called angiotensinogen, and basically upregulates the pumping of blood, upregulates blood pressure, and then upregulates something called the prorenin receptor, or PRR. And PRR is a component of the RAS, and its receptor is renin. And PRR has a soluble form, and it's called sPRR, which I will be focusing on today. So some goals for my project was to study the tissue of origin of increased circulation of sPRR to see if it's actually in the adipose tissue, so in your fat tissue, and whether sPRR is actually contributing to obesity. And so my hypothesis for this was that the oversecretion of human sPRR, just to make it more local to us, in the adipose tissue induces obesity hypertension through the activation of the RAS. So now for my methodology and how I did this experiment. I took 8-week-old control and cream mice, and there were about 5 per 8 mice per group, and there were 4 total groups. And I fed them a low-fat diet for the span of 0 to 16 weeks. And so yeah, I had Cree, male and female low-fat diet mice, and control, male and female low-fat diet mice, which is a total of 4 groups. And now for my model validation to test if my actual model works. I just basically crossed a male Cree sHPRR with a female sHPRR, which had a silence gene, and it created my Cree mice, my experimental mice that oversecrete sHPRR, and my control mice, which don't secrete sHPRR. And then I did some genotyping to figure out which mice were my experimental mice and which mice were my control mice. As you can see in figure A, all the bands at the top just basically means that the primer binded to some DNA, which shows that I had DNA. And then the second level was basically all of the ones that had a black little line means that they're the experimental mice. And then all the ones that didn't mean that there are control mice. And I did this by taking previous experimental and control mice and loading them in the qPCR plate, and then comparing that to my experimental mice, or my sample mice. And I had a total of 8 experimental mice and 7 control mice. And then I did some validation just to test the cell growth throughout a few days, just to see if sHPRR was secreted, and if my cells were growing regularly. And as you can see in the graph, they were, and we're seeing what we expected, that the Cree mice, as you can see throughout the days, had an increased level of sPRR, which were all significant when compared to day 0 and the control in their different days. And then I did a model validation on the epididymal fat tissue, the sHPRR levels in the epididymal fat, which is just an adipose tissue. In females, it's perigonadal fat, and in males, it's epididymal fat, or EF. And as you can see, there is an increase in sHPRR levels, signifying that sHPRR is actually released in the adipose tissue, like we predicted. And then my final model validation was just to test if sHPRR did go into the circulation. And in this case, it wasn't a significant increase statistically, but it did increase in males and females, signifying that sHPRR does contribute to the circulation. So now for my actual results. After having these mice on a low-fat diet over the span of 16 weeks, I did the body weight evolution. So in graph A, you see that there are, it's a 15-week period, and graph B is at the end of the 16 weeks. And this is for females. So as you can see, the graph didn't really increase and stayed steady throughout the experiment. And then in graph B at the end of the experiment, you kind of see that there is also no change, signifying that this doesn't occur, that the body weight due to the oversecretion of sPRR doesn't really affect body weight in females. But now in males, the body weight evolution over the span of the 16 weeks increased significantly. And as you can see in graph D, at the end of the 16 weeks, there is an increase in the cream mice mass, signifying that sPRR does contribute to body weight evolution in male mice. So now the results of my body composition. So as you can see, this is the female mice. And I did this through an eco MRI, which basically tested fat mass, lean mass, and total water. And as you can see, again, you're seeing that same trend. There is no increase in body composition in comparison to Korean control mice, signifying that in females, there's not a increase in body composition, that sPRR does not affect it. But now in males, again, there is a significant increase in the body composition. So fat mass, lean mass, total water kind of shows where sPRR is being increased throughout the entire mouse. So this shows that sPRR affects the whole body of the mouse in fat mass, lean mass, and total water mass. So body composition, the oversecretion of sPRR does have an effect on body composition in males. And then I took the weight of an adipose tissue, the EF and paragonadal fat that I was talking about earlier, to kind of pinpoint where this is occurring. And as you can see in female mice, there is no increase like we've been seeing, but in male mice, there is a significant increase in the adipose tissue weights at the end of the experiment, signifying that sPRR does contribute to the overall weight of the tissue and makes it bigger, which does contribute to obesity. So in conclusion, the oversecretion of sPRR increases body weight, body composition, and fat mass in male mice, but we weren't seeing it in female mice, which could be because females and males are really different throughout kind of all animals, so that could be a factor of it. Yeah, so the body weight composition, fat weight, was all increased due to sPRR in obesity. And this signifies again that sPRR does have an effect on the overall increase of body composition, body weight, and fat mass in all mice, or just male mice for now. And so for future research, I will be testing a high-fat diet in Korean control, because I've been seeing this increase of body composition, body weight, and fat tissue mass in a low-fat diet in male mice. So it'd be interesting to see how much more it increases in a high-fat diet when obesity is actually going to be put into play. And then I will also maybe put the mice in metabolic cages to kind of show that how much the mice are actually eating, because in this case, some mice could be eating more and some mice could be eating less, which could contribute to my data, and a metabolic cage would take that into consideration. So yeah, and now I would like to acknowledge the people that's helped me throughout this project. My mentors, Audrey Poupou and Kelly Nichols, both are great in their line of job. And then Gertrude Arthur, who is a graduate student, kind of helped me with my training and how to do certain things around the lab. And of course, the lab director and research director, Dr. Lea Yiannikouris. I would also like to thank the University of Kentucky and the Kentucky Young Researchers Program for letting me be involved in this great experience. And I would like to thank my friends and family for support over the span of this project. So yes, that is my project. Thank you so much for listening to it.